ठीक है सर, स्टार्ट हो गया। लिंक मिला आपको चालू में? ये स्टार्ट हो गया।
ये वाला ना,
ये
ठीक है ना,
यही है सर।
ठीक है।
Send it to me, I will send.
ये साउंड भी जा रहा है सर। ठीक बोलिए सर, प्रोसीजर मालूम है ना सर आपको?
एक मिनट,
It is that?
उसके कोई डिस्क्रिप्शन में कोई आता है, ऐसा होता है डिस्क्रिप्शन में कोई कैंसर।
Okay, it didn't change it. Hello, Mr. Rohit. I'm Dr. Saikaran, SR Microbiology. Hello. I have sent the link for practical class today. Yeah, I have sent you the link for practical class today. Yeah, you forward to your colleagues. Okay, right. Good afternoon, everyone. Today we have a practical demonstration for molecular techniques in microbiology. I'm here to show some of the instruments and articles that are used in our molecular techniques. So the molecular technique that is used in microbiology is PCR, polymerase chain reaction. As you know, it's a technique to obtain multiple copies of specific DNA fragments, that is target DNA, from the samples. Even a very meager amount of DNA is also sufficient to multiply, and it is mainly used for diagnostic purposes. Here, coming to the basically there are three different steps of PCR. Before that, we need to have something called master mix that has all the constituents required for the amplification of target DNA in the sample. These essential components are: first, the template DNA, that is the DNA that contains the target sequence of interest. Then primers. Primers are defined as a pair of synthetic oligonucleotides that have complementary 3' ends of each 2 strands of target DNA. So for us to initiate the amplification process of target DNA, we should have a starting codons. Those are called the primers. These are synthetic oligonucleotides. Oligonucleotides are nucleotides of 5 to 18 number. They will initiate the process of amplification. Then other things that are needed for amplification in the PCR mixture, that is master mix, is the thermostable DNA polymerase. This is an enzyme that catalyzes the amplification of DNA from the parent strand, that is template strand. So this acts with like DNA dependent DNA polymerase. So from DNA we can amplify that DNA. So from DNA we are amplifying the product DNA. Then the other things are the divalent cations, that is magnesium ions, that are required for the activity of these polymerase. Then deoxyribonucleotide triphosphates. So D and TP. So these are the main building blocks for DNA polymerase activity, with which the new strand is formed. Then finally, the buffer solution is needed to maintain the osmotic balance, ionic environment, and stability of this polymerase. Here, this is the instrument, that is PCR. It's called as thermocycler. Here we will be putting the tubes. So before we first we receive the sample in our lab, we are going to first extract the DNA from the sample. Then that extracted DNA is put on for PCR. So what is the procedure for extracting the DNA? So this is the DNA extraction method, where we have something called spin column. Okay, here we have a silica. Here, this is called as spin column, where this is used in the extraction procedure. Here we have some all the DNA that is extracted get filtered in this silica, and from which we can take the desired amount desired DNA. So this is called as spin column, that is used for extracting the DNA from the sample. So here is the flow chart for extraction. So for the release of DNA, then for formation of a column, then we add the solution ethanol and we spin it. After spin it, we will have all the DNA that is entrapped into the silica. Then we have to wash it and spin it for 1 minute, and we will be eluting the DNA from this. And regarding the steps of PCR, basically they contain 3 steps. First is denaturation, then annealing, then extension. Here the temperature is very important. First, the denaturation, the sample is heated to greater than 90 degrees centigrade, where the 2 strands of DNA will get separated. Then annealing is the process where the temperature is lowered on 3 to 5 degrees centigrade, that allow these primers, oligonucleotide primers, to bind the single strand of our target DNA, then initiate the process of amplification. Then this follows the extension where the final step where the temperature is typically raised to 72 degrees centigrade, allowing specific enzymes like DNA polymerase to act and form the template, from the product DNA. So there are many instruments that are used. Here, this is the flow chart where, as I said, first they get denaturated. Two strands are separated over here. Then we will be cooling it and annealing, where the primers get attached to the specific sites. Then on the other side, the amplification has got started, and you can see the extension where the DNTPs are added one by one, and you can finally get a daughter DNA where you have two complete strands. In this way, this process happens, and this is one cycle. Then the second cycle starts where again each of these product DNA will get denaturated and primers get attached and extension happens. In that way, the amplification happens exponentially. So during this process, we are going to use many instruments. So here, this is the pipette. This is very important instrument where we are going to handle this minute amount of all the samples or liquid, all the DNA that is extracted. All these are very minute quantity. We are going to use this pipette along with the pipette tips to transfer this elute or the product or amplified product from one place to another place, or one chamber to another chamber, or to during store, like this is the PCR tube. I can show. This is the place where the extracted DNA is placed, and this is put into the thermocycler for amplification. So before that, I'll be showing, this is the pipette. This is the pipette tip with which we take all the desired lipids. And there is one thing called this. There is a filter here in the pipette tip. Okay. The pipette tips are put at the normal type and with a filter in the unit. This filter will prevent the contamination. These are called ependoffs. These are used for aliquoting. In the sense, we can store the sample or the amplified product in this, and we can store at desired temperature. These are the PCR tubes where we can load the extracted DNA and we put in the PCR machine for amplification. So these are the plates, PCR plates. This is 96 well plate. This is 48 well plate. You can see. This is the bottom part. This is the upper part. So we load the extracted DNA and we put it on the thermocycler for amplification. So after amplification, the next process is detection. So how do you detect whether the desired quantity, DNA is amplified or not? This is done by the conventional method, the gel electrophoresis, where you can see here, this is a buffer tank, where you will be placing the gel over here. Here in this, the gel mainly consists of 2% agarose powder used for gel preparation. Then we use a dye called as ethidium bromide. This is the intercalating dye. This dye will attach to the DNA double strand. So DNA separated on the basis of charge and mass by applying electric current in the buffer solution. In this buffer solution, we apply current here. This is the positive pole. This is the negative pole, where where we apply this current, and by applying this current, this depending upon the charge and mass ratio, the amplicon will be, the amplified DNA will be moving along each comb. And after that, we will be putting, then we will have a ladder over here. Ladder is a reference where we have already mapped the target DNA of interest, the mass and mass of the target DNA. If this is used for matching. So we can see whether if our desired mass, if you are detecting a pathogen, if the mass charge ratio, according to mass charge ratio, if this is the location, if the DNA can move to this location with the help of this, like by referring to this ladder, we can diagnose that our amplified product has is significant. So there are many methods of, this is the conventional method where we see the detection of our amplified product depending upon the mass charge ratio. The other modes of detection are the real-time PCR and where we use the fluorescent dyes. That will I'll tell in the further slides. So what are the variations of PCR? PCR can be of many types: reverse transcriptase PCR, multiplex PCR, nested PCR, and real-time PCR. The meaning of reverse transcriptase PCR is basically the template will be RNA. So RNA is first converted to DNA by reverse transcriptase enzyme. Then this DNA that is formed is called as complementary DNA, cDNA, and this DNA goes through the normal process of DNA amplification, and with which we can detect, we can diagnose. Then what is multiplex PCR? Multiplex PCR, we can here we have many targets genes, and we are going to put all this PCR at once. So for different targets, we have a specific different primers, and we put all the master mix, and we can add the sample. So with the single sample, we can try to diagnose. With a single sample, we can try to diagnose multiple sets of primers and different diseases at once. So single sample, we can try to diagnose multiple diseases with multiplex PCR. Then what is nested PCR? Nested PCR, here we have two sets of primers. So one set which covers a broad segment of DNA. Then within that, there is another PCR that is put. The amplicons that are the product that is during the first PCR run, the product that is formed that acts as a template for the next PCR. So to understand, first there is a wide segment of genome that is selected, and the PCR is done, and within that there is some more smaller segment. Okay, that this PCR is nested within this broad PCR. So this is called nested PCR. Then the last is real-time PCR. Here in real time, that says that the PCR, the amplification and the detection can be seen at the same time. That happens with the help of the fluorescent probes that I'll discuss later. So in real-time PCR, here this is the PCR curve. So this is the real-time PCR curve where we can see a graph. So these fluorescent probes that are used in this detection. So you can see here in y-axis, you can see the change in the fluorescence. Okay, the delta R, that is called as the fluorescent signal that is generated during the PCR process. Okay, that is shown in the y-axis, and the x-axis shows the cycle number. So the main meaning is, as the DNA start amplifying, the fluorescence gets generated. So the amount of fluorescence that is generated is directly proportional to the amount of amplification that is being done. Here in y-axis, I can, as I said, it is the intensity of fluorescent signal, and x-axis number of PCR cycles. So this is a threshold value. This is a reference value for calculating the CT value, and this is a baseline. What is baseline? Baseline is the fluorescent signal that is generated before the actual PCR, the amplification starts. So finally, this is called the CT value. What is CT value? It is the cycle number at which the fluorescent generated within a reaction crosses a threshold is called that is CT value or threshold cycle. This CT value is inversely proportional to the quantity of template DNA. The meaning, the DNA before this is the signal that you can see before the actual amplification gets started. Then at a single cycle at which the fluorescence that is generated, that shoots up and crosses the threshold value, this is called as CT value. CT value is a cycle number. So where to apply this? As if you have a sample suspecting a virus, if the viral load is very less, then the CT value will be more. If the viral load is more in the sample, the CT value will be less. That is, very less number of cycles are needed for it to shoot up and cross this threshold value. If the viral load is more, CT value will be less. Why? Because very less number of cycles are necessary for it to cross this threshold. Crossing the threshold in the sense fluorescence increases exponentially. That indicates the amplification has started and the exponential growth is taking place. So this is the example for real-time PCR for influenza virus. This is how the machine looks, where we can see this is how we are placing the PCR plate over here, and we can see real-time graphs in the screen. So what is melt curve analysis? So in this, generally cyber green is a dye. This is a non-specific dye that binds to every double standard DNA that is amplified during the PCR process. The other part is the other dyes are the probes. These probes are specific for specific DNA. But whereas this cyber green is non-specific, then this cyber green, after the real-time PCR amplification, in this melt curve analysis, the machine program is to perform melt profile to ascertain the specificity. So as I said, cyber green is non-specific. It binds non-specifically. To see the specificity, we are going to do the melt curve analysis. So what it says is all PCR products of a particular primer should have same melting temperature, unless there is a contamination or mispriming or primer dimer artifact. So this is melting curve. So this says all the PCR products, this mainly indicates the specificity. All the PCR products for a particular primer, that is primer of interest or the target of interest, should have the same melting temperature. Here in this example, we can see a red line. These are all these lines show these all belongs to the same target. But this red line shows, see here, all the sample runs with the same primer pair, but the sample that contain no DNA, or that is the red line, shows a melting curve lower than the TM, the threshold, compared to the other sample. This is probably due to the primer dimer artifact, or since there is no DNA, that is the primer dimer pairing happens, and that shows in the form of a red line. So the other variants of this PCR are the GeneXpert. Mainly in our laboratory we do for MTB, and also we can nowadays we also do it for detection of SARS-CoV-2. So not only this, there are many places where we use GeneXpert. So basically GeneXpert is it is based on the principle of real-time PCR. It's a rapid molecular detection system, and coming to the tuberculosis, it can detect within 2 hours the tuberculosis, and also here we can also detect the rifampicin resistance by targeting RP-OBG. Okay. So it requires no instrumentation other than this system. This is the instrument GeneXpert, where we have modules over here. In each module we can fit this cartridge. So this is how the cartridge looks. So this is GeneXpert cartridge, where we can place a sample over here below this lid. This is the dummy cartridge, where we can this only for demonstration. So the invalid cartridges, we are going to put the sample over here and close it, and we are going to scan and give the ID number, and we have to put this into the module of GeneXpert system. So here first the sample washing, extraction of DNA and RNA purification. So in GeneXpert everything is in a closed container. There is a minimal chance of contamination, and here this is a place where we can put the cartridge, and basically this works on the basis of real-time PCR, and it is fully automated. So coming to the next, BioFire FilmArray multiplex PCR system. This is the most advanced and recently developed system where the sample preparation, amplification and detection all three run in a single instrument. Here in this instrument we have all the things in a single place. These tubes are the reagent storage system. Then this is a chemical circuit board here, where the first part, this is dedicated for sample extraction and sample preparation. Then with the help of vacuum, the material will move from one part of the circuit to the other part of the circuit. Then this is the place where the first stage multiplex PCR happens over here. This is a sort of nested PCR. Here the cyber green is used. Here non-specific detection occurs. So this is the first stage of multiplex PCR. Then after amplification premiere, they are transferred to the second stage PCR. Here in this second stage, there are many panels. Like each panel has a different system. For example, there is a respiratory panel, gastrointestinal panel, or meningitis panel, where we have list of all the organisms, let it be bacteria or virus or any other organisms that are that can cause, for example, if you take respiratory panel, all the organisms that cause respiratory infections are included in the panel. And here I have given an example where this has, you can see adenovirus, Bordetella, respiratory syncytial influenza, metapneumovirus, chlamydia pneumoniae, parainfluenza, rhinovirus, H1N1. All these things, all these organisms are included in a single panel, and each dot represents the different targets, and all are detected at once. So this is the most recent and advanced, and for demonstration this picture is more clear. And in our lab we have here, as I said, here this is the place where the sample is loaded, sample processing. First, I think you can't see over here, it's totally transparent. First set of PCR, then second set of PCR. Here this is what I said, where the panel of organisms are included. This is it. So finally, coming to nucleic acid hybridization, mainly this is based on the ability of nucleic acid strands that pair up with the complementary base sequences. This forms a duplex or a hybrid. Here in this we have one strand that is known, one strand that is unknown. The known strand is probe with the defined codons and everything, whereas the unknown is called as a target, where in this nucleic acid hybridization process, one strand, the probe that originates from organisms or nucleic acid sequence with known identity is is sent on the target with unknown. So these probes can go, for example, if we have a sample with unknown etiology or with unknown organism, these known probes can, if they are sent into that, they will go and bind the complementary sequence. If the complementary sequence and binding happens, then the fluorescence, we can see the fluorescence that gets originated. With that, we can say that it is used in the detection. So the application based upon these are the line probe assay. It identifies Mycobacterium tuberculosis and drug resistance. DNA microarray disease diagnosis and drug resistant, gene profiling, drug target identification. These are all the applications of nucleic acid hybridization. Then fish fluorescent in situ hybridization. It's also for diagnosis of different diseases, and blotting techniques like detection of DNA, RNA, and proteins, etc. So this is nucleic acid hybridization. So these are the basic molecular techniques that are used in our laboratory, and these are all instruments that are used for the purpose. Thank you.